Give comparison between the following: C3 and C4 pathway, cyclic and non-cyclic phosphorylation, anatomy of leaf in C3 and C4 class. So students, let's learn the differences between these two. First, let's take C3 pathway and C4 pathway. Now, C3 pathway occurs in plants like wheat and C4 pathway occurs in plants like maize. So, both pathways is related to photosynthesis. So, when photosynthesis takes place, the first stable compound formed in C3 pathway is phosphoglycerate. So, we call this pathway as C3. And in the second one, the first stable compound formed is oxaloacetic acid, which contains 4-carbon compound. So, we call this pathway as C4 pathway. So, let's find the difference between these two pathways. So, in C3 pathway, the carbon dioxide is accepted by a 5-carbon compound, which is called as RUBP. The atmospheric carbon dioxide, the Rubisco enzyme fixes the atmospheric carbon dioxide, and this is accepted by a 5-carbon compound, which is called as RUBP, Rubisco bisphosphate. Whereas in C4 pathway, the carbon dioxide is accepted by a 3-carbon compound, that is phosphoenol pyruvate. So, phosphoenol pyruvate will accept the carbon dioxide. In the C3 pathway, once the carbon dioxide is accepted by RUBP, this is unstable and this splits and gives rise to a first stable compound, which is called as 3-phosphoglycerate. So, the first stable compound is formed is 3-phosphoglycerate. This compound contains 3 carbon atoms. So, based on this, we call the pathway as C3 pathway. And this reaction occurs in the mesophyll cells of the leaf. So, here we can see these are the mesophyll cells. So, in these cells, the reaction takes place. And in C3 plants, the chances of photorespiration is more. So, here carbon fixation process is less and the chances of photorespiration will be more. So, the rate of photosynthesis might be affected in C3 pathway. If we come to the C4 pathway, the first stable compound formed is oxaloacetic acid, which is made up of 4-carbon compound. So, first the carbon dioxide will be fixed by the mesophyll cells. In the bundle sheet, oxaloacetic acid is formed and then this oxaloacetic acid will be transported into the bundle sheet. So, here Kelvin cycle takes place. So, the Kelvin cycle occurs in the bundle sheet, whereas in C3 pathway, the Kelvin cycle occurs in the mesophyll cells itself. So, this is one more difference. And this fixation takes place in the mesophyll cell and bundle sheet of the leaves. And when compared to the carbon dioxide fixation, the C4 plants fix atmospheric carbon dioxide more faster when compared to the C3. And here, the chances of photorespiration is low and the rate of photosynthesis will be more. So, these are the major differences between C3 and C4. The next one is cyclic and non-cyclic photophosphorylation. So, cyclic photophosphorylation occurs in the photosystem 1 and it includes only one photosystem, one reaction center will be present. And this involves synthesis of ATP. So, here photophosphorylation takes place and ATP is synthesized. In this process, the photolysis of water, that is breakdown of the water in the presence of light, releasing oxygen, electrons and protons. So, this process does not occur. So, oxygen is not produced during cyclic phosphorylation. And in this process, electrons move in a closed circle. That means the electron which is lost will come back to the reaction center. So, we call this as a cyclic photophosphorylation. So, here we can see when the light is absorbed, the chlorophyll gets excited and it releases the electron. This electron travels and energy is produced in the form of ATP and then it returns back to the reaction center. So, we call it as cyclic photophosphorylation. And here photolysis, that is breakdown of water, does not take place. Coming to the non-cyclic photophosphorylation, Now this has got two reaction center, photosystem 1 and photosystem 2. This involves synthesis of ATP and NADPH2. So both ATP and NADPH will be synthesized. And this non-cyclic photophosphorylation, there is photolysis of water. The water will be split and it releases oxygen which is liberated. Along with that, protons and electrons are also released. And the electron which gets excited from the reaction center doesn't come back to the reaction center back. So in the reaction center, chlorophyll are present. When it absorbs light, it releases electron. So this does not come back to the reaction center. So we call this as a non-cyclic photophosphorylation. So this is a non-cyclic photophosphorylation. So here we can see the light is absorbed, the chlorophyll get excited, and then this excited electron will be transported to the next photosystem, and during the process both ATP and NADPH will be released. Now NADPH will accept the electron, so it is not coming back to the reaction center. Meanwhile, water gets split. When the water splits, electrons and protons are released and even nascent oxygen is released. This oxygen will be released into the environment. Electron which is released by the water goes to the reaction center. So that is why we call this as non-cyclic photophosphorylation. Photophosphorylation means formation of ATP. So in both ATP is formed, but NADPH2 is formed only in non-cyclic photophosphorylation. Okay, the next one is anatomy of leaf in C3 and C4 plants. So let us observe the leaf anatomy of the plants C3 and C4. In C3 leaves, they have upper and lower epidermis, stomata is present, and the middle portion is called mesophyll. Now the mesophyll have got pellucid and spongy parenchyma. In between vascular bundles are present, and vascular bundles are surrounded by the bundle sheet. So bundle sheet cells are present, and in C4 plants, upper and lower epidermis are present, and the mesophyll cells are arranged in circle layer. So there is two circular layer. So one is the bundle sheet cells, which is closely arranged, compactly arranged without intercellular space, and this is surrounded by another layer of mesophyll cells. So both C3 and C4 have got bundle sheet cells, but the difference is in C3 plants, the bundle sheet cell does not have chlorophyll pigments, whereas in C4, the bundle sheet cells have got chlorophyll and chloroplast. So the chloroplast is present both in mesophyll as well as bundle sheet, whereas in C3, the bundle sheet does not have chloroplast. Thus the C3 leaves, the mesophyll cells in the C3 leaves, they have rubisco enzyme. So rubisco enzyme is present in the mesophyll cells. The first stable compound formed is 3-carbon compound, that is phosphoglycerate, and hence it is named as C3 plants. And in these plants, photorespiration takes place. So here we can see the atmospheric carbon dioxide enter into the mesophyll. The rubisco enzyme fixes the atmospheric carbon dioxide. The first stable compound formed is 3-phosphoglycerate, and this process is called as Kelvin cycle. So Kelvin cycle occurs in the mesophyll. Now, when the carbon dioxide concentration decreases in the mesophyll and oxygen concentration increases, the rubisco enzyme acts as a oxygenase. So photorespiration occurs in the C3 leaves. So, and this can affect the rate of yield in the photosynthesis. Whereas in C4 plants, the rubisco enzyme is present in the bundle sheet. So the rubisco enzyme will be present in the bundle sheet and absent in the mesophyll cells. And therefore Kelvin cycle occurs in the bundle sheet and not in the mesophyll cells. So the first carbon compound formed is oxaloacetic acid. Later it is converted into malic acid and stored. And when photosynthesis takes place, the malic acid will be transported to bundle sheet, and here decarboxylation takes place, releasing carbon dioxide. So this carbon dioxide will enter into the Kelvin cycle and the food will be synthesized. So always there will be availability of carbon dioxide. So therefore in C4 plants photorespiration does not take place.